Topic complement system is actually a set of around 30 plasma proteins which function or which complements the functions of antibodies in combating the microbes, etc. So going on to the topic, so why should we know about it? This is a system that is protecting us on a daily basis from a variety of pathogens, and there are many disorders that are associated with either the hyperfunctioning or the hypofunctioning of the system. A few examples being hereditary angioedema, PNH, atypical HUS, and recurrent infections, of course. We'll come to some of them later. Now, in a complement system or a pathway, there are three steps basically: the initiation, amplification, and termination. Amplification is mainly done by the alternative pathway, and termination is when a membrane attack complex forms and the cell gets lysed by that. Now, based on the initiation, we can divide the system into classical pathway, alternative pathway, and the lectin pathway. Now we'll go on to the classical pathway, or no, sorry, the concepts. Some concepts that we have to know before going into the topic. So these complements are mainly not mainly, they are proenzymes. That is, I told they exist as plasma proteins, that around 30 of them are there, but they are also seen in the interstitial fluid, not just plasma, also seen in the interstitial fluid. They are proenzymes in the sense they are not active by themselves. They don't cause any activation or destruction in their native state, but when they get activated, they can form the, or they can continue the pathway. And how they get activated is when they get split into two components by proteases. There are enzymes called proteases which can split a protein, and when these proenzymes get split up, they will form two components: a smaller fragment and a larger fragment. Smaller fragment is A, small a, and the larger fragment is B. You can remember as B for bigger, because the larger is B. And once this proenzyme gets activated, they can further continue a cycle, like they can cause a chain of reactions by, once after they get activated, they themselves can act as proteases and continue a chain of the cycle till the final membrane attack complex is formed. So that is what they get activated by proteases, and they themselves act as proteases once they are activated. Now, by convention, the complement is denoted as C, and in case of classical pathway, it is numbered complement 1, complement 2, complement 3. That is based on the order in which they were discovered. And in case of alternative pathway, the nomenclature is a little different, in that we can have a capital B, D, P, H, and I. In this, B and D are the active proteins. P is properdin. Properdin is basically a stabilizing factor. So P is a stabilizing protein. H and I are the inhibitory proteins or brakes. That is, they prevent the overactivity of the alternative pathway. They act as brakes. Now, this is just an example of how this proenzyme gets activated. C2 is a classical pathway. By convention, you can see the C2. It was the second protein discovered in the classical pathway. When C2 is cleaved by a protease, it will form C2A and C2B. C2A is the smaller fragment and C2B is the larger fragment. And below you can see the capital B. That is the protein that you can see in the complement pathway. And capital B will form two fragments again when it is cleaved by a protease into capital B smaller, that is BA, and larger BB. Now, how does it all work? This complement, once they get activated, they can either act as anaphylotoxins, they can act as opsonins, or they can form membrane attack complexes. Now, what is anaphylotoxin? Anaphylotoxin is this can recruit neutrophils and other inflammatory cells. It can cause leakage of blood vessels, increase the permeability of blood vessels. Opsonization means this can form opsonins or bind to the surface of the pathogen, and in that way it will help the macrophages in recognizing that there is a pathogen out there, go and attack it, eat it up, or whatever you want to do with it. Then it can form membrane attack complexes. Membrane attack complex is a pore-like structure. The proteins like C5, C6, like about 4 of the proteins, C5, B, C6, C7, C8 and C9, sorry, 5 of the proteins, they form a circle. Just imagine as the chain of proteins forming a circle, and they will form a pore, and they will attach to the surface of the pathogen, and this pore will allow the exchange of ions and water in and out of the cell, and ultimately water will get inside the cell and the cell will get hydrolyzed. Opsonin, the most common opsonin or most important opsonin is C3b, and in case of anaphylotoxins, the most important anaphylotoxin is C5a. Now coming to the classical pathway, the first protein is C1 discovered, and C1 is actually a complex of proteins in that it contains 3 other proteins. It is when 3 proteins come together, that is what we call as C1. That is C1q, C1r and C1s. In this, C1q is the large protein, C1r and s are smaller proteins attached to this larger C1q fragment. And when C1q binds to the constant region of the Fc region of the antibody, which is attached to another, or which is attached to a pathogen, an antibody attached to a pathogen, to the constant region of that antibody when C1q binds, or if C1q binds to the pathogen by itself, C1r, the small fragment, gets activated, and the activated C1r will cleave the other smaller fragment, that is C1s. The C1s is a protease when it is activated, and the C1s will cleave another protein, C4. C4 is cleaved by C1s into C4a and C4b. Now I have told B for bigger, so C4b is bigger and heavier. Just imagine like that, it is heavier and it will stay on the surface of the pathogen. C4a is quite light and it is an anaphylotoxin, and C4b will attach to, so C4a is an anaphylotoxin, C4b is C4b will attach to the surface of the pathogen, and when C4b attaches to the surface of the pathogen, C2 will attach to C4b. Remember, all these proteins are freely floating in and around the plasma. So this freely floating C4 was cleaved by this C1s, and C4b was formed, and once C4b formed, it got attached to a nearby pathogen surface, and when C4b was attached to the pathogen surface, it saw a C2 which was again moving around in the plasma, and when it saw that a C4b has bound to a pathogen, it came and attached to this C4b. So now we have a complex C4b, C2. Now another property of the C1s is that it can again act as a protease on this complex of C4b and C2. Please write it down, it's quite complicated and it is going to get more complicated. So C4b, C2 can again be acted upon by C1s. So C1s will again act on or is again going to act as a protease, and it will cleave the C2 into C2a, the smaller fragment, and C2b, the larger fragment, and the larger fragment will stay. So we have a new compound, or a new what do you call clump, that is C4b, C2b. Now C4b, C2b is called as the convertase or C3 convertase of the classical pathway. In any complement system, in whichever pathway you may say, you may think of either of the three, the formation of C3 convertase is the most critical step in any of the pathways. So the most critical step has occurred, that is the formation of C4b, C2b, which is the C3 convertase of the classical pathway, which has formed on the surface of the pathogen. Now this C3 convertase can act as a protease and divide C3 into C3A and C3B. Now C3A acts as an anaphylotoxin just like C4A, and C3B will act as an opsonin. In fact, C3B is the most important opsonin, and it will attach to the surface of the pathogen, and it will give signals to the nearby macrophages that, see, I have a opsonized cell or a pathogen. Now you can come and gobble it up or whatever. Now coming back to the convertase, now the C3B, so we can see that C3B can act as an opsonin, but C3B can also attach to the previously formed complex and form C5 convertase. So C3B can independently attach to the surface of the pathogen and act as an opsonin, or it can attach to C4B C2B complex and form C4B C2B C3B complex, which is called as C5 convertase. Now C5 convertase will act on the C5 in the plasma and will divide it into C5A and C5B. Now C5A is the most important anaphylotoxin. Now that is about the classical pathway. Now we'll go to the MBL pathway. MBL pathway is very similar to the classical pathway. In this, it is also called Menin binding lectin pathway, or just the lectin pathway. In this, the MBL is a protein that is similar to C1q, and MASP1 and MASP2, that is serine proteases 1 and 2, are like C1q, C1R and C1s. So the function is quite similar, but the thing is that the MBL will attach to only the mannose residues on the surface of pathogen, and when MBL attaches to the mannose residues on the surface of the pathogen, it will get activated, and then just like in the classical pathway, all the steps occur, and finally C4 and C2 will be cleaved by the MASP2. Now this is the view of the Calicut Media College night view taken during a program. Now coming to the alternative pathway. Alternative pathway is, I have already mentioned, is the amplification part of the whole among the all three pathways. Alternative pathway is the one that amplifies all the three pathways, including the alternative pathway. So alternative pathway can amplify MBL pathway, it can amplify classical pathway, and it can amplify itself. Why is it so? This is because the C3 convertase and all is different in the alternative pathway. It is an alternative pathway in that there will be spontaneous hydrolysis of C3 that is seen in the plasma. Now again, C3 is the most common complement protein in the body, and it is seen in the plasma and interstitial fluid. So spontaneously, some of, or a very small fragment or fraction of C3 will spontaneously get hydrolyzed, and that is called C3H2O. This spontaneously hydrolyzed C3 or C3H2O, it has the same function as C3B. C3B can act as opsonin, if you remember. Now the C3H2O, you can consider it as C3B because it has the same function as C3B. Now it can bind to B. B is an alternative pathway protein, an active protein, and D is a protease in the alternative pathway. When C3H2O or C3B gets attached to C3B in the presence of the protease D, the B protein can get cleaved into BA and BB. The BB will attach to the C3H2O or C3B, and that is called the C3 convertase of the alternative pathway. So C3H2O and or C3B will, in the presence of D protease, and when combined with B, and the B will be fragmented into BA, smaller BA and larger BB. The BB will again attach to the C3H2O or C3B, and that is what you call the C3 convertase of alternative pathway, C3H2O BB or C3B BB. So you can see here, this C3H2OBB or C3BBB will act on the C3 that is again floating in the plasma, and it will form C3A and C3B. C3B is again an opsonin, if you remember. Now the C3BBB that I have told is the C3 convertase. It can or it will bind with protein P, another protein in the alternative pathway. It's also called properdin, and then it will stabilize. This properdin will stabilize the C3 convertase. Actually, this alternative pathway was previously known as properdin pathway because of this, and so it will be stabilized. The stabilized C3BBB can attach to another fragment of C3B, and that can form a complex called C5 convertase. So again we have different converters here. In case of alternative pathway, it is C3BBB, C3B. Okay, it's getting confusing. Now C5 can get acted upon by C5 convertase and form the anaphylotoxin C5A and C5B. Now coming to the point, C3 convertase can be C4B, C2B, that is in the classical pathway, or C3BBB, that is in the alternative pathway. C5 convertase can be C4B, C2B, C3B in case of classical pathway, or C3B, capital BB, C3B in case of alternative pathway. Now the C5B can attach to C6, C7, C8 and C9, some of the other complement proteins that are seen, and it will form around a ring-like structure or a pore-like structure, and this pore will attach to the surface of the pathogen and a pore is formed. We'll come to that. Now the anaphylotoxins, we have already told C5A, C3A and C4A are the anaphylotoxins, of which C5A is the most important one. Opsonin is the most important one, is the C3B. Now another thing that you can or you may want to know is C3B can be hydrolyzed into inactive C3B or IC3B. Inactive C3B can still opsonize. It doesn't lose its opsonizing function, but it cannot take part in the amplification process. That is that is why it is called inactive C3B. It can still be a good opsonizer, but it cannot, or opsonin, but it cannot amplify the pathway. Now membrane attack complex, which I was talking about, C5B to C9, it is a set of proteins which will form a circular structure, a pore-like structure through which the fluids and ions can move in and out. Ultimately fluid will move into the cell and the cell will get lysed. This is a houseboat from my state, Kerala, small state, South India. Okay, now some of the disease associated. Now C1 inhibitor, that is another protein which can inhibit C1R and C1S. So C1 inhibitor will inhibit C1R and C1S. So what will happen? That means classical pathway will be inactivated. But if there is a deficiency of C1 inhibitor, there will be overactivity of the classical pathway, and that patient can have a recurrent attacks of severe hereditary severe edema. It is called as hereditary angioedema. That is so C1H, C1INH or C1 inhibitor is deficient in that condition. Now CD55 and CD59, of which CD55 is also called a decay activating factor. These are the proteins that are linked to GPI anger. GPI anger is a protein that links to the surface of the RBCs, and to this GPI anger protein, CD55 and CD59 is attached. And in case this decay activating factor CD55, it prevents the formation of C3 convertase. That is the normal function, and the normal function of CD59 is it inhibits the formation of membrane attack complex. So in case of PNH, which is an acquired disorder, there is a deficiency or a problem with the functioning of GPA anger. So when the GPA anger does not anger to the surface of RBCs, the CD55 and CD59 also cannot anchored to the surface of RBCs, and so there will be overactivity and destruction and lysis of the RBCs ultimately. Now coming to deficiency, those deficiency of membrane attack complex, or any defect with the membrane attack complex formation. In that case, there will be recurrent attacks of Neisseria or Neisserial infections. Then if there is any deficiency of C2, C3, properdin, factor D, etc., there can be a recurrent pyogenic infections. And in case of any abnormality of the, or in case of abnormality of the alternative pathway, we can get atypical HUS, hemolytic uremic syndrome. Why it happens is like this membrane attack complex, when there is abnormality of alternative pathway, there can be increased formation of membrane attack complex, increased amplification, increased production of membrane attack complex. This membrane attack complex can also destroy the endothelium of the blood vessels, and when the endothelium gets destroyed, the subendothelial area gets exposed, and that can lead to formation of thrombus, fibrin, etc. And the deposited fibrin can act as scissors, and when an RBC passes nearby, this fibrin will act as scissors, and it will take up a part of the RBC membrane, so we can get schistocytes that we see in the peripheral smear. And also when the subendothelial layer get exposed, there can be increased formation of thrombi, etc. That is atypical HUS. Thank you.